make sure there's no question on that. Yeah. So, so this is, now this might be where some of you know it all and others not. So I don't know exactly, you can maybe wave me on if it's too familiar. And I'll start at very, very basic things. So we're talking about the brain, right? So the nervous system generates cognition, as obvious, right? And the brain, the brain is this thing, right? And the emphasis I want to make is it has a visual front end, the visual cortex, the general sensory cortices. We'll often talk about vision as sort of the main source. And that has a motor output at some point, right? The motor cortex does project down to the spinal cord and does do some motor stuff. And it has, the other thing, like the frontal cortex that does some, goal-oriented stuff and so on, very roughly speaking. And, this language of saying it does that and so on, this is really just labels we put on something, right? Doesn't really explain how that works. It's just very short language. I'll say a little bit more about that in a moment. And the brain consists of neurons, right? A lot of neurons, 10 to the power 11, maybe even 12 in humans, and about 10,000 synapses each. There are different kinds of neurons. Sometimes interesting to keep these in mind, right? There's this, a lot of the old connections models, every neuron had like 5 synapses. So, now you may know that on average, again, there are different types of neurons, but for a lot of cortical neurons, the input of about 10 spikes that come in through the tree and that overlap enough in time, like 10 milliseconds next to each other. That's typically sufficient to fire a neuron like that, right? So that is 1% of the inputs, right? That is maybe something that is far from what a lot of artificial neural networks do. On the other hand, 10 to the 12, sounds like a lot, but I'll show you later an example where when you extrapolate what we're doing, you very quickly run into this limit. You quickly need more neurons than there are particles in the universe if you are too naive. The combinatorial problems of having a neuron for every combination of everything, that's the binding problem actually, can very quickly escalate. So it's kind of useful to keep in mind that there are limited resources. And the brain is a very costly thing in higher primates and humans, including humans. The brain is a major limiting factor. You might know that some of our primate friends have to spend a lot of time just eating because they need to get all the energy for their brain. And there's some speculation that humans really need fermentation or cooking and fermentation to be able to optimize the energy input so that we don't have to spend all time eating. Orangutans, for instance, spend 10 to 12 hours a day eating. They eat a little less valuable food that takes more effort to then actually eat. So the brain consumes a lot of power, and there's a lot of evolutionary pressure to minimize that. That's why I believe that's sort of underestimated how much of neuroscience is really about that, about optimizing, minimizing the energy. The spiking is probably optimize the sparsity. And the sparsity is real misunderstandings out in the field that some of these principles, for instance, optimal coding, which predictive coding, which is used in all kinds of misuse in all kinds of contexts, is very likely something that is really to minimize the effort for certain tasks. And we use that actually in technology to where it's sort of functionally invariant. That is, for instance, when you use predictive coding in, for instance, video encoding, the video quality is supposed to not depend on it, right? So it has no functional significance, it just reduces the footprint of what you're doing. And so these things are very important in the brain, and this is not what we're trying to do here, right? This is something, so that's why we have to sort through the neuroscience literature and find the functionally significant parts of it. And by function, I mean for cognition and behavior, right? And separate from that, other problems the brain has to solve, right? It has to develop, grow, and be plastic, and have homeostasis. And the fact that we do brain imaging, as many of you know, reflects that, right? The brain imaging is possible because there's such good control over the blood flow to to keep the oxygen there, that we can just take that for granted and then take it as a functional signature. But actually, it means there's a lot of machinery to do that. We have about as many, actually even a little bit more astrocytes, for instance, than we have neural cells. Astrocytes are involved in these kind of processes. So just, there's a huge thing here just to say why it's not trivial for us to pick out the neural principles. Okay, so neurons are also very variable, but they all share this structure that they have sort of an input-output structure, right? They have the dendritic tree and ultimately output through the axon, and at some point near the soma, spikes are generated. That's sort of the input-output schema, even though it differs in different neurons. Now, when people talk about brain function, they primarily use these techniques from coding, from communication theory. So coding is really just a fancy way of saying there is a relationship. A code is just an invertible math, right? And what coding really means is that you have something inside, you measure something inside the brain, vary something outside, and if there's a dependence, then you say the inside codes for the outside. If that dependence is invertible, then it's technically correct that you do that, but sometimes people even gloss over that. It's just some functional dependence. I worked in a neuroscience lab in Marseille, and my colleagues always use that word, and then we had all kinds of misunderstandings. And someone said, "What you mean by that, this coding, really just that the firing depends on that, task variable." And they didn't like that, but yes, that is actually what they meant. Right? And in fact, in neuroscience practice, when you are in the lab, that is really what you as a scientist, what you do. If you've ever been in a neuroscience lab, once you have an awake behaving animal that does its task, and you have your electrodes in there, you put them on a loudspeaker, you hear these firings of the neurons. You put it on the loudspeaker because you're playing with your electrodes to capture the right neurons. And the right neurons means the neurons that are somehow related to the task. And that means that when the animal does this task, that they fire always at a certain point in the task. And maybe that also, are sensitive to exactly which task the animal does. And that means their firing depends on that task. And you call that then coding. But so you're actually you're all tuned to only look for neurons that have that property. If there's a neuron that doesn't do that, then you move your electrode somewhere else. You're not going to record that neuron. Right? So when people say there are all these neurons in this area, this means that's the ones they recorded. There are other neurons there that don't modulate their firing with the task, and then you don't record them. Right? So this coding idea and the decoding idea is the more mathematically exact form of how to test whether you can predict the condition under which the neuron was recorded from its behavior. That's sort of the core of all of that, and doesn't really say anything about function. So we will be using this concept because we will be saying that the fields and so on have a certain meaning, and it is in terms of that coding of input-output relationships that they have. Neuropsychology is sort of the inverse, right? There you're changing the substrate and then seeing what that does to the behavior. And optogenetics is now the holy grail of that you could just find in a fine way lesion brains and so on. This is some idea, inside, outside. So here you're changing the inside and then seeing what behavior emerges. In the other case, you do the behavior and look what happens inside. Both of these have a hard time with the kind of activity that we'll talk about that is autonomous, when it's actually not triggered from something outside. See, this kind of technique doesn't really do much for that, right? But so here's a little bit a roadmap of what I can talk about. So very briefly about neurophysics, and by that I mean just the dynamical equation of the membrane, and then talk about rate code, receptive fields, maps. The only original stuff is this distribution of population activation and arguments for population code. And the last 2 points are also just conventional. So that will be the only original part of here. And the others I can go through very quickly, I presume. Right? So neurophysics all I want to say is the membrane has that differential equation. I don't know if you know that the membranes, all the neurons are electrically charged. They have a potential difference to the surround. Ion pumps keep that actively around. It's all well understood. Hodgkin active would be maybe the model that's the most fundamental one here. And they all amount to having this kind of equation. So if the membrane potential is U, then U dot equals minus U is sort of a fundamental linear equation that has a stable state at U, the right-hand side being 0, and the resting potential when there's no external input is then a certain number. Here it will be, actually, I don't think the cursor works when I'm in this. Yeah, I can do that. Yeah, good. So here will be like 10 millivolts. And for instance, if in a neuron you insert some electrical shocks, which will be the very direct way of putting input there, then you see the exponential time courses. You can directly estimate this tau, and that's about 10 milliseconds, depends on the kind of neuron. But that's of the order of magnitude of what we have there. It's very slow, right? If you think that you can do behavior in 150 milliseconds would be one of your fastest reaction times. And 250 is quite reliable. You can do movements as fast as that, And the elements in there, their time scale is only, a factor of 10 faster. Compare that to computer, where it's like 10 to the power of 15 faster than the time on which we are using it, right? So that's one interesting challenge, actually. So we'll be using this year. We'll be using this idea of time scale and this fundamental mechanism, right? The minus U this year is not a neural network kind of equation, right? It is a physics type of equation. That is, this would be, you couldn't interpret this minus U as a coupling of some form. It changes sign depending on U. So it's really physics. It's membrane physics. Then, of course, in spiking neurons, there's threshold behavior. The threshold behavior is spiking. If you have a large enough input, you get a spike, an action potential, time course that has its own, is actively generated, and that is something that we're not considering. Spikes, there's accumulation of inputs to make a spike, and that idea is summarized just in the sigma. Yes, please.
Sorry, they're not equation. What would H represent?
So H will be the resting level. So if there's no external input, then in a sense, just a calibration what the meaning is. So physically, it's in actual voltages, and H sets that level. Later, we will be using abstract activations where we set the 0. We can define what it is. In fact, we do that exactly with respect to the threshold, right? The spike, the threshold is some voltage, depends on the neuron type. We will be using universal variables in which we replace the spiking by a threshold function that only makes sense across populations ultimately. And then we can set arbitrarily where that threshold is. We set it to 0. So negative is below threshold, positive is above threshold. And so U isn't really the membrane potential anymore. It's some kind of activation description. And later we'll use activation descriptions of populations. In our modeling, we actually use something homologous to the membrane potential, which and as contrasted to the spike rate. A lot of people think of activation in connections network as being spike rates. And that has the downside that you don't have a good expression of what happens sub-threshold, because it can be very deep sub-threshold or close to sub-threshold, and there's a lot of difference about that. And the spike rate, you can do things like spontaneous firing rates, and then you're a little bit below that, but it's very difficult to represent that. And in the brain, there is a representation of how deep below threshold you are, because the membrane potentials are there, they're real, right? So we're using a description. There's sometimes computational neuroscience talk about two different pictures where you either have the presynaptic, the pre-threshold description or the post-threshold description. Mathematically, you can actually transform one into the other, but it's the picture we're using is the one where we're dealing, would be the pre-threshold one, that is we're formalizing the membrane potential. But it isn't literally the membrane potential, right? It's some kind of microscopic description of that. And the sigmoid is actually how that generalization is done. So that eliminates spiking and replaces this by that. Yes, please.
Are we always going to look at spiking neurons? Because I know there are some that just function completely without spiking.
Yes, so we are actually not looking at spiking neurons. That is, we're eliminating the spiking in our description. And I mean, in computational neuroscience, people wonder about that, right? There are these non-spiking neurons. And so the fundamental question is, do the spikes actually have any functional significance? Or are they just an implementation issue, right? Saving energy. And that is an open question. Many people model everything in spiking neural network just because there are spikes, and then say, oh, you have to understand how it works with spikes. There are some cases where there is a suggestion that special functions could be enabled by spikes. For instance, anything to do with synchronicity. What you could say, the spiking mechanism makes that the networks are sensitive to timing if the spikes overlap enough. And I had this here. So the summing up of postsynaptic potentials to make a spike, the inputs have to be close enough in time, about 10 milliseconds, because of the membrane potential time scale. So after 10 milliseconds, everything has decayed from previous spikes. And so that means that your neurons are good at, or would be good at detecting temporal ordering. And that has given rise to ideas like that, synchronicity as a form of binding, for instance, that's clearly debunked now, I would say. But in other, for instance, echolocation would be the most famous examples really done in owls, for instance, to see how they compute time differences. And then this spike timing plays a role. So I should say I was in a lab in Marseille where that was a major agenda, and we did population code, and they with the same data they did spike timing, which was a real nuisance because they did a lot of trials to do that. We wanted a lot of conditions, and so we had a compromise, which was bad for both sides. Because it's very expensive, with an animal, it only behaves for 2 hours, you only get this many neurons and so on. You can see it's always trade-offs. And so my summary, it's an interesting story because my colleague Alexa Riele published a science paper there about functional significance of spike. And now she's actually no longer confident that the paper holds. So I was not involved in that. So I think that my summary would be we've never really established any functional significance of the spike timing. And so that's a debate, I would say. So if that was true, there was actually no real functional significance, then it would be no difference. It's just an implementation issue. So that you would do non-spiking where it's got to be fast and it's crucial, like very close to sensory surfaces. It happens in insects, for instance, for very fast circuits that they have, and where the energy cost isn't so important because it's very critical what to do. And then when you have more luxury type of activity like cognition, which is sort of an add-on, then you want to save energy and do it by spiking. Roughly speaking, as you have very large brains, and we know myelination, there are a lot of tricks to get this stuff going. I have these long cables across the brain, how to transfer activity. That's a real problem, and that's what spiking could do. Right? So this is the most radical view that I espouse that pretty much. I could be wrong, right? It's not what we're really working on. It's just the assumption we make. Yes.
Maybe it's a moment to ask, but beyond what about the rhythms that are constituted through those spikings? Is there any kind of functional role attributed to that as BFC?
No, actually, I was asked that question in Rotterdam. No, I think that many of these rhythms, these better rhythms, gamma and so on, these high frequency rhythms are possibly just signatures of this implementation starting with these high frequency events, and they have no functional significance. And if they seem to have functional significance, it's only because there are population states in which there is more or less activity, which does lead then to those changing. But this is a major problem when you do spiked timing in neurophysiology, which I know very well because of this work back then. Because when the spike rates change, the degree of synchronicity changes just because by chance the spikes will have to be closer to each other when you have higher frequency. Right? So it's actually complicated to separate synchronicity from rate, because changing the rate will change synchronicity just by statistics. And so you have to have more synchronicity than that to say it plays its own role. So I don't actually see any connection. There could be some different theta rhythms could, if they're slow enough so that they are on the time scale of behavior, then they will play a role. And in our kind of thinking, active transients or population level timing, I'll actually talk about that. Population level timing is the way time courses are generated. These are time courses on the order of time scale, order of magnitude of hundreds of milliseconds, the kind of things that you do in movement and that you see in certain cognitive operations. So they are not these high frequency time signatures you see in the EEG and things like that. But this is a somewhat radical position. So and it's not our main thing to reject that kind of thing, right? So we don't really know, but we don't see. Yeah. Patterns of activation that when we talk about it. Yeah. So we mean by that macroscopic population level patterns, and they are activated. Essentially, our modeling is only fixed points, so stationary states, and these slow oscillations, which are in the functional range, so in the range of movements.
And the rhythmic properties are just some kind of iffy phenomenon.
So, again, it's a question what rhythmic means. So we'll have a little bit of lecture about motor behavior. And in motor behavior, which we often study in a rhythmic sense, I originally come from movement coordination. And it actually turns out that rhythmic movement is a sequence of discrete movements, which in some sense, because the back and forth is actually driven by different systems and they alternate. So in the brain is actually an alternation, but you could still say the whole thing is maybe a rhythm, but it's more a sequence really. So it's discrete, but it has well-defined time courses. I like to talk about timing, that is it. Well-defined time courses, but it's usually just an active transient, do it once, and then something else triggers the next one and so on. And then, of course, it can look rhythmic if you just repeat that. And so currently in the motor area, maybe that's what you're referring to, people like Churchland and so on, talk about neural oscillators and so on. I like the conceptual idea, but actually I think the work is totally misleading. So it's a bit ironic. I like the direction, but I think it's not a good argument, because the only thing is that they have the monkey do a periodic task, and then you see periodic activity in the brain. And there's nothing special about that, right? Yeah. So we have to, can go into more detail about that. Yeah, interesting discussion coming up here. Yeah. Good. Okay, so that's all I wanted to say about neurophysics. So, it's of course a huge field where there's a lot more detail about these time courses. And we just check from that u dot minus equals minus u. That's what we'll check from that. And now sensors. So that's the periphery, and the periphery is the eye, the ears, and so on. You can send it. Roughly speaking, there are only really three kind of sensors. It's the photoreceptors, it's chemical receptors, and everything else are hair cells. Some bending of something, some elastic thing that makes spiking. And then in the ear, you have that hearing is like that, the skin, everything. So a lot of these hair cells in different forms. You can't even see here the details. Let's just put that up to illustrate the image. So those are the kind of sensor cells. And the sensor cells are about transforming something physical into something neural, right? That's what we sometimes talk about rate code there, in the sense that it's sometimes an invertible relationship between those things. That is, you stretch one of those, I don't even remember what cell it is, some stretch receptor in the skin, let's say, and then you get either a potential or a firing rate, depending on the kind of sensor, that is an invertible function of that. So you could say if there is more firing, it must have been more stretched. And so you could estimate from the rate the physical state if it was calibrated. There are all kinds of sensors like that. I don't know, some other thing. Photoreceptors are actually quite complicated things. And if you look into the retina, it's kind of mind-boggling. I have actually a damaged retina, so I'm very sensitive to this now. It's even, I don't want to go into the details of that. We often use a very simplified picture of that in our model, right? So in the periphery, there is some kind of rate code, right? So meaning that the function, the firing rate or potential is, co-varies or, is a function of something physical. And later we will say there is more something called space code, where the firing is just saying that whatever that neuron is responsible for is present, but the firing is more an internal thing. It doesn't really represent anything outside. On the motor side, it's similar, right? Ultimately, there are neurons that connect on the muscle, and when those neurons fire more, then the muscle contracts more. So for instance, here's some of these motor neurons, and here's how much they are firing, and then how much, I think, force or whatever it is, can't even read that, some physical measure of what the muscle does, co-varies with that, and there are different kinds of muscles and so on. With muscles, it's already a little bit more complicated, because a muscle that doesn't have a reflex loop isn't really functional. So the stretch reflex is the most elementary one, where it's a monosynaptic, right? There's a receptor in the muscle that sees how much it's extended, and then when it's extended more than a threshold, then it will activate the muscle to resist. And that makes it a dynamical system, as a recurrent kind of loop, and a dynamical system that has an equilibrium point, a fixed point, which would be the preferred length of that muscle. And when you have posture, then that's what you actually have. When you have posture, it means if you're perturbed away from the posture, you are firing more to resist that. Muscles only work in one direction, right? The other ones are then not stretched. And when you lose that, deafferentation is done in the lab, but there are also some people who are unfortunate enough to have a disease of the certain sensor cells, and they don't have this working. Your muscles are totally useless. It's a big myth in the lab that you can learn, and monkeys can learn to make some movements, but it's totally non-practical. It's only, you can actually train monkeys to still make some movements when they're deafferented, but they do it very differently. So some patients who suffer from that are totally helpless, and they cannot even sit, and they will have to look at their body to not fall over, even in a chair. Because actually any posture, anything that you do is stabilized against physical forces, against any perturbation. And if you don't have that, which the reflex does, you don't have that stabilization, the muscle doesn't have a well-defined state. So that's an interesting complication. So even though you say the force is a function of the input, actually what you're doing is you're setting a stable trajectory, it's a fixed point, stable fixed point for that peripheral system. And if you think about it more deeply, that's actually true about the sensory side as well, especially here in the retina. This is a real issue. In the sensory side, you're all familiar with that. If you have a sensor, that sensor will be, we say it will only be responsive to certain frequencies. Right? It will have a preferred time structure. And that's because it's actually also a physical system that is coupled to the incoming physical signal. And that is a dynamic system, therefore, and that dynamic system property matters under some circumstances, right? So that's just to say the rate code is sort of an approximate description of that, and we use that in our theoretical framework ultimately to map something physical. That, for instance, we take a video image and say that's our vision sensor, and then we will take the for instance, the saturation of a hue channel, which is not really physiological, but we take that as something that is translated into activation. That's where we're making this assumption. Okay, so that's rate code at the peripheral surface. So we'll simplify that very radically. Now, the other thing is, of course, you have networks. This is an example of the retina. As it's already doing a lot of complication. The output of the retinal ganglion cells already have this invariance under contrast, right? So this is here, the blue sky is probably 6 orders of magnitude brighter than any structure in the room, but you can actually see both, and that's at the same time, and that's pretty amazing. And if you have a camera, you know, the cameras are much worse. Even though the iPhone's not very good, they imitate some of that. And so that is very much done in the peripheral nervous network. I just say we gloss over all of these kind of things, and this is a problem because there are interesting normalization problems of the nature I just hinted that are solved there, and we used to be quite bad with that. We're now much better. That is, you know, quite specific machinery that's really, it's been sort of like the limit of the kind of things that we describe. As we go very much into the periphery, our approximations break down ultimately. All I want to say is that the receptive fields, right? You're maybe all familiar with receptive fields, right? That means that there's a certain range of stimuli that a neuron responds to, and we can track it through the neural architecture from the retina to the LGN to the visual cortex, centers around. There's certain time courses on, you know, so rich literature on that. And ultimately, here, let me take the Hubble and Wiesel. This is from the original paper, the tuning curve. So first of all, only a certain range on the visual, on the retina is where when light falls there, the cell in visual cortex will respond, and then it will respond more when there's a moving light bar that moves in certain directions across that receptive field, and the tuning curve is a function of the orientation and movement direction of this bar. Is a very nice function. That was, you know, the fundamental discovery about the visual cortex, and that is how we will define the feature dimensions that we'll talk about, right? The forward connectivity that makes these kind of tuning curves. Receptive field is just the tuning curve for space, right? It's the same kind of idea. Receptive field profile would be to say within the receptive field, how much firing would there be for a dot of light in there? And the analogous thing in the motor system is that you can have some neurons in the motor cortex that fire differently when you make different movements. So famously, different movement directions. You would have neurons that fire more for movement directions of a certain hand in space, right? A certain way than others. And I'll come back to that in a moment and talk more about that. So that's how that's where coding arises, right? And when you have a neuron that depends on, the firing of which depends on the stimulus condition or the motor condition, then we say the neuron codes for that. And all that means is there is this input-output connection. And the theoretical account is it's the forward network that makes that. And that's an idea that we're using. Okay, that's that. And then in the brain, when you systematically map that, you find these feature maps. That is, you find that then that the tuning curves are sampled, all the different neurons sample the possible tuning curves. And in some cases, you have what's called topography, that is neighboring cells have similar tuning curves. That's really what it means, right? So for instance, you have a retinotopic map. That means that the neighboring receptive field on the visual surface are in neighboring locations in the brain. That is really just that has no functional significance as such, right? It really just means that the cables are not too twisted, right? And it's probably a result of the growth of this tissue. And there are feature maps that are not like that. For instance, in the motor area, you have that there are cells that code for a certain movement direction, and then neighboring cells code for a different movement direction. So it's just more scrambled there. There are other constraints for that system. These topographic maps, yes, please.
If you're always, and from your experience, is that if I have two cells and they react in completely different directions of movement, is there some logic as to why they are still next to each other, like some higher level reason that they are like that, or is it just or do they just get assigned based on energy?
I don't think this is known. I mean, in the motor area, there was a long effort to find topographic maps, and there are some topographic maps in the sense, for instance, of which muscle groups are coded where. So that, and you might have seen this motor homunculus, that there would be certain areas that are for the fingers and for other parts. that actually is probably a distortion. There is some work by someone called Graziano, if anybody knows this work, has shown that actually you have to know how these are made. So these maps were made by stimulating motor cortex electrically and then seeing muscle twitching somewhere. And it turns out, now this stimulation was very brief, because if you do more, then you burn the tissue. And then you get this homunculus. But if you, there's some techniques of how to stimulate for longer without burning the tissue, which Graziano came up with. And so you stimulate a physiological time, which is like 200-300 milliseconds. And then you find that they code actually movements. So for instance, there we have finger twitching. That was actually a curl movement where the hand to face movements is elicited. And the first thing is the fingers curl, and then everything else follows. And so it just looked like it was about fingers. But then when you stimulate longer, it's actually about this whole movement. And so that says that it's a failure to confirm this topography. But there is a sense that there might be a topography for movements, but we don't really know. It's just a limitation of knowledge. It's much easier to find order than to prove there is no order, right? But it's just important to see that that isn't really functionally significant. It becomes functionally significant in what we'll do, where we talk about interaction. That is, we'll be postulating that neurons that code for similar things are coupled differently than neurons that code for dissimilar things. And then and if interaction is a function of distance on the cortical surface, then topography suddenly has meaning. And I think that's likely actually really what is at play here. But we'll actually not really worry about how these things are mapped onto the brain. And I'll show you in a moment that population code gets rid of that as a major determinant. But I'm assuming that you're all familiar with this kind of maps, right? Have heard about them. And they have this property, if you have a map that is not just 2D, now a retinal surface would be 2D, and then you have an orientation or color and so on, then you cannot consistently map that onto 2D. So you have to have piecewise maps, right? And there are some signatures for that where like an orientational map has to then reset every, like you have those partial maps and you can figure that out. And sort of the idea is that these maps would have limited dimensionality. That is, if you think how many things are coded there, it will be maybe 4 or 5 parameters, but not 50, which would be difficult to map onto a 2D surface. Again, so this is there's a lot of emphasis on this, and we take from that there are these feature maps which will not bother about how they are arranged on the cortical surface. We'll just span them in the function space, in the stimulus space, and similar for the motor maps. I don't even talk about the motor maps because they're too complex. Now, okay, so this is textbook stuff that you hopefully probably already know. I just remind you of that. Now let's talk a little bit about this idea of distributions of population activation. So first of all, population activation, I say outright, our claim, or what we took from the literature is a claim that population activity is the best correlate with behavior. Now, that is, I would say there is consensus about that, even though there's still some, neuroscientists like to talk about differences and so on. But, if you have a rough picture, I would say that is currently the consensus in the field. And now we have to be a little bit more specific about what we mean by that. And the idea is that when you have a tuning group, let's say you have neurons that are tuned to some parameter, I don't even remember which one it is, orientation here, differently. This one prefers that direction, and the different neurons prefer different directions, which will mean that if you show this population one particular orientation, then one neuron will fire a lot and other neurons will fire less, right? And so one simple picture is to say, oh, only the one that fires a lot, that's the one I will do winner takes all, which is the theoretician's way of saying I pick, just one neuron, and I will take the preferred direction of that neuron to be my current estimate. And that's a lot of neural networks do that, and they have an algorithm for doing it. So the winner takes all is actually some arc max, right? You find which firing, you look up, the address of the neuron that fired most. And so population code would be the idea that all the firing matters. So the neurons that fire a little bit will also contribute to your estimate. So you're not throwing away any neural activation, which would make a lot of sense if it's so costly to fire, right? And so that's an empirical question. Is that actually the case? And there is, in the meantime, very solid evidence that that's the case. I show you the historical work by David Sparks' group this is actually colliculus, but sort of like subcortical, but has very similar principles. The saccadic movement system. So that, make these abrupt eye movements. That's called saccades. And has a sensory area, motor area. We don't need to go into that. Let's say in the motor area, you have a topographic map where you are essentially encoding saccadic endpoints. So if you electrically stimulate somewhere on that cortical on that map, then you will elicit a saccade of to a particular retinal location. Actually, depends on the starting location as well. And so I could say, for instance, that if I stimulate here, I get this saccade. If I stimulate here, I get C isn't even shown here, maybe A here. I get that saccade and so on. That will be the map. And you typically have broad tuning. That is, when I do a particular saccade, a whole region, there's a little activity bump on that map. And so what these people did is they were able to reversibly deactivate some of the population. So for instance, if you have this saccade to B, then this region here, the red circle, would be where neurons are active. And now you could say, well, what if I can deactivate these neurons while the animal is moving, animal is trained to make saccades to certain stimuli, while the animal is trying to make a saccade to B. And if I can do that, if I deactivate that, and these neurons do contribute, even though they are not the most firing neurons, the saccade should be deviated. It should go more in the direction of D than in the direction of C, because those are voting for, for the saccades closer to A, those are voting for this B, and those are, coding for D. And so it should be deviated from B toward D. And that's what you get. So rather than to go to B, the animal then makes a saccade that's off. And that shows that these did contribute after all. And it's reversible. They can actually they do it by cooling. They have a little electrode that cools the neurons. And when they are too cold, they don't fire. But then when you stop cooling, then they fire again. So they don't kill the neurons. And so you can really show it's really caused by that. And there's been similar work now on, for instance, MT, an area that is for detecting visual movements. And so the monkeys have to estimate which movement they saw by, with the joystick, they indicate the movement. And again, you can sort of bias the perception by taking out different parts of the population. So that's actually uses topography, because now you can, in a targeted way, deactivate these neurons because you know how they're arranged. In motor cortex, for instance, that's not possible. That's historically a little bit ironical, because historically, these data from motor cortex were considered evidence for population coding. But there, because of this mixture, you cannot, in a targeted way, deactivate some neurons because they're all mixed up. And the argument here was simply that the tuning curves are so broad. Like this is 360 degrees, right? So in all directions, you have a cosine tuning curve. So the neuron is firing pretty much in all directions, but a little bit more in some. And then the argument of Jacopoulos was, well, that must mean that it all contributes. And the proof was just that by taking these curves into account, he called that population vector, he could predict the movement. So he recorded in some condition, and in other conditions, he was able to predict the movement from the population vector. And that was an argument why it should be population code. And actually, we have a variant of that I'll show you now that does that well. I have a variant here for visual cortex first. Let's maybe do the motor first, because it makes more sense now. So this is the work we did in Marseille, actually, where we did Jacopoulos type tasks. Monkeys make pointing movements, in particular directions. It was like on a vertical surface in that case, and there's the center button, and then they move to one of these only six targets, and there's some the targets changed color to tell the monkey when to make the movement and so on. In this experiment, we manipulated some prior information, like it was either 3 targets or 2 possible targets or 1, which had some purpose. And so these neurons fire preferentially for certain directions, and they fire actually mostly before the movement is initiated. The movement is initiated here at this point here, and that varies, and there are all kinds of complications about that. And you can actually now construct what we call a distribution of population activation from the firing of all those neurons. So the way one can do that, there are different ways, and this is one possible way, is to take the tuning curve of that neuron under a certain kind of condition, like the condition where the monkey knows exactly which movement it will have to make. This is a caricature, right? These green curves would be the tuning curves of different neurons. And to estimate the population distribution, you would take those as basis functions. Every neuron contributes that tuning curve, and you multiply it with the firing of that neuron under some other condition, some new condition. So for instance, if the condition is just one of those things you use in determining the tuning curve, the neurons that have the tuning curve centered on that direction would fire a lot, so they get multiplied with a lot, and so they would be making this large contribution, while the tuning curves, that are far from that would be multiplied with less, and they make a smaller contribution. If you add them up, you get this curve. That would be the distribution of population activation. And with that technique, so that's it, rather than look on the cortical surface, right? This is looking in movement space, in the target space, and you use the tuning curve to span that space, right? The tuning curve is a function over the movement space, just movement direction. So distribution of population activation is in the functional space, in the visual or movement space, not on the cortical surface. And so with that, you can do a lot of things. I mean, it's kind of trivial that it would have these properties within the population of responses that you had when you established the tuning curve. But now you can use the firing rate in a new task. So in this experiment, we use just in certain time windows. We get the tuning curve from like the whole episode of firing, and then we can look at different time intervals and look at the time course of the activation. And then we were also looking in these intervals when there was just some prior information. And other people, and I'll show you another result, looked at like decision-making tasks. So here's a estimation in time that we took as current firing rate, always just, I think, like 50 milliseconds, or I don't remember, 20 millisecond time slices. So you can see the evolution of the population activation. But here toward the response moment, you see firing increase. In fact, the monkey is actually the tuning curve, the population peak starts to rise before the response signal is given, because it's predictable here by the monkey, so it actually anticipates that movement. You see a little peak here. That peak is the preparatory signal. When the signal comes in, the monkey knows which movement, and already the neurons are firing a little bit more. And we were able to show that depends on how broad the range is. It tracks the range. And if you have a broader range, then you have a broader peak, and it predicts reaction time and all kinds of stuff. And here's a very nice example from John Kalaska's group, Paul Zizek, who used the same technique, even though they say cell here, but it's actually the same technique. It's not cells, it's the tuning curves that span the space. And so this is in dorsal premotor cortex, where you kind of see selection decisions. I say this because we'll literally do that later with the field. They actually modeled this with our kind of fields. This was after we published that work. So for instance, in this task, the monkey initially has the initial position of the hand is here in some center, and then it gets a cue that there's a red target in one direction and a blue in another. And in different trials, these are in different directions. And then it has to keep that in mind. And then there is a color cue, which tells monkey which color target to take. And then there's a go signal, and it's only these directions that ever occur. That's how you get the tuning. So in this case, this here would be where the monkey has to move. And using this population representation, there's about 100 neurons that contribute to that in different sessions, right? And then you build this distribution. You see that in an early phase, they represent both possible targets, and that's somewhat sustained through the delay period. There's sustained firing here in this premotor area. And then when the decision comes in, one of those gets boosted and the other gets inhibited, right? It's below resting level, and that's sustained. And when the go signal, it also predicts the go signal somewhat. And then when the go signal comes, this choice gets activated, right? So you see, this is a way to take neural activity and look at it functionally. And that's really what the fields do, right? The fields are, this would be the neural estimation of fields. Now, only a few cases where we really did this at the neural level. It's a huge effort to do that, and you're very constrained by what animals can do. But it's really quite consistent. I wanted to show you this other example here first. We historically did this first here in Bochum. Dirk Janko, who has a research group here. This is when I was first here before going to Marseille. And this is, unfortunately, anesthetized cats, area 17, so primary visual cortex. And just receptive, just retinal space. So you do, there you have these receptive fields, right? And so you first compute you record different neurons, right? You have to put the electrodes in. It's actually across animals. It's going to really take different recordings from different animals and align them. You align them by having always the same spatial sample where the retina the fovea would be here. So you have a fovea-centric space in which you vary the stimuli. And the critical idea is, like for the monkey, that you would now use just the same stimulus for all cells. Right? In the monkey, that makes one movement for all cells. Well, classically, people always look at the receptive fields, the tuning curves, so they give stimuli that are different for every cell, that are centered on its receptive field. And the whole literature consists of lists of properties of cells that would be like that. So when you do population, that's why it was discovered in the motor system. Right? In the motor system, it's very natural that the animal has one task and you're measuring cells. You're not giving a different task for every different cell, right? And that's why they were able to see that there are these broad activity patterns. While in the sensory areas, they would always give specific stimuli for every cell, just where the cell fires. We didn't do that. So we determined the receptive field in the classical way, just to mark every cell, but then we presented stimuli to all cells. So these spikes are where cells fire in different amounts. So we had cells that fire here, so where there's stimulus here, was quite far from the receptive fields. So we get, no, I should say the other way around. You say there are cells whose receptive field lies here, and we recorded them even under conditions where they wouldn't fire, right? That's why nobody does this historically. And that enables us now to pool the responses of all the cells into a distribution of population activation. So we did that by some techniques, so you filtering and then superposing. And so same idea, the receptive fields profile is the tuning curve, is the base function, and then you scale that. There's some linear optimized stuff and so on. So it's a bit more complicated than I'm saying now, but that's the core idea. To get smooth things like that, you have to do some filtering. And so here, just to show that that works, these are the stimuli that we show to all cells. So different is a little square of light that is at different locations across the retina. This is roughly foveal space, so this will be still the foveal area ends around here. So you see the peak of activation tracks the symbols, so pretty much good estimation of retinal position. And now you can again do this game of inserting different current activity under different tasks into that system. So here is this the one? Yeah, this is the one where we have a complex stimulus. The complex stimulus was just 2 squares of light, very simple. So the 2 little white things, you see here's a white thing and there's a second one. Here, maybe here you see it, right? There is a white square here and a white square there. So these were 2 squares of light that were flashed on and off. And so we vary where the second square is. And what you're seeing here is the actual response on top to that stimulus. And here on bottom, the superposition of the 2 responses to the individual when you showed this separately. So to compare, essentially look for interaction. That is, does the response, is it just the sum of the individual responses, or is some interaction between those. There is, of course, for instance, if they're very close, the sum would be larger because you might have some threshold, right? Oops. So the fact that you have less here than there could be just that the neurons are threshold and you don't fire twice as much when you have the second stimulus on top. But when you move here to the far end, they are not overlapping. So you could imagine from the previous picture, the left, the right, if you add those, they don't overlap. So this here is really the response to one stimulus and that response to the other stimulus. And now you see that when you show them at the same time, you have less fire here. That is actually what we call global inhibition or inhibitory interaction. That when they're presented simultaneously, they're weakening each other. It turns out there is also excitatory interaction that is facilitation in here if we get rid of this problem of hitting the ceiling, and that can be done with some techniques. So it's a one-dimensional cut. And for instance, here's an example. There's always the superposition with the dashed line. The action response is the solid line, and you see here some excitation. That is, there is more response to the composite stimulus than to the individual stimuli added early on. You only see that excitatory interaction comes in early because it's mediated by the same neurons. I'll explain that a little later. So we have just showed that because we have some real neural evidence for local, that's neighboring in stimulus space, excitatory interaction and global inhibitory interaction at longer distances. And that's what we'll be postulating for all fields. Because, this is just an anesthetized cat, area of 17, just one little thing. It's a huge effort to even get that. So that will be ultimately how we would aim to get evidence for any kind of our postulates, right? And again, that exploits this fact that we have the same stimulus for all neurons. Yes.
As a narrow basis for attention.
Well, yeah. I mean, yeah. I mean, actually, almost everything else. I mean, this is of course very, very early. That we'll be talking about in visual cognition is essentially the thing that you call attention. And Raul will be talking about that for 3 hours. So in many of the models that I'll be talking about, the visual representations are actually the attentional system, not this very early system. Okay, so these are, this is, yeah, one can also do that in other spaces. Maybe I'll skip that. And so maybe that's enough about this here. Yes, please.
Because I had to include these receptive fields before. Was the temporal receptive field also included this computation? Because there's always also a curve in time for the receptive field. Like there's a shape in time of the stimulus that they refer. I mean, I did the rational variable set specifically.
That is true. So if you so this is an old story that how do you establish? So if you stimulate, then there is a volley of activity in the cell. So it has a time course. Actually, in visual cortex, this is about, for instance, about 70 milliseconds after the stimulus onset. And let's say if you have a one-time onset of a stimulus, you have a volley from 70 to about 120. And also it's different anesthetized, non-anesthetized. And there is very little sustained firing actually, which is a real problem. It's a very transient phenomenon. And so for a long time, people have taken, this includes us, I was here actually when we worked forever on time-dependent receptive fields. So you can either say there is a receptive field that characterize what the cell stands for, and now I look at the time course of activation, that's what we do. Or you say the receptive field itself is time varying. And that will be an idea that what the cell represents is some spatial temporal filter. And there were other people who tried to do that. There is no right or wrong thing, it's just how useful is it, right? These are just, it's just us, it's our instrument, right? These diffusions on, that's like your thermometer, right? If you have a thermometer, the thermometer as such doesn't do anything. It's just a way of how you connect the length of the liquid in the tube to temperature. And there's no right thermometer, it's just a question of which one is more useful. So this is a tradition to consider space-time receptive fields. I don't think that it buys you much. It really only buys you something if you go into essentially a certain kind of systems theory. Of course, my former boss here, who was the first the director of this institute, was very much in that line of work. I don't think it's useful. I think it's more useful to say I'm characterizing the forward network by some feature spaces, and then all the time courses are interpreted as a reflection of the neural dynamics, right? Be aware that it's just, and that will be important in DFT, we'll be saying this is a field that represents orientation or represents, whatever it is. And that is just for us, the scientists, to remember what this field is about. It's a label we put on, because really, ultimately, it's just some neural dynamics going, and ultimately drive some muscles and moves, right? In our case, of course, the motor front end is often very simple. And that this activity somewhere has this meaning, that's just a guidance for us, right? But it doesn't have to be, it has no fundamental meaning as such, right? It's just some way how we orient in this big dynamic system. So we think of the brain as this big dynamical system. We'll use this particular framework with fields, and it's only the behavior in the end that matters, right? The insight, that is more for us to have some survey. And so for that purpose, we think of static receptive fields and time courses as informative about the processing, a better picture than spatial-temporal filters as of the elements. Does that answer your question?
So we can just do without the temporal part, and
No, we capture the temporal part by the dynamics of activation.
that's interesting.
Yeah. Rather than so it's where you could say the receptive field is a projection operator, abstractly speaking, right? And that is, you're now taking from something from a neural space that you're sampling, and you project it onto something physical, for instance, orientation of a stimulus, or some color or something. And therefore, you're projecting that activity. If you have 100 neurons, so you're projecting that from some hypothetical neural space into a parameter space. Mathematically speaking, if you knew the original space, you could write down this projection operator. And for that purpose, we believe it's more useful to have static spaces, feature spaces, and so on. And say all the time courses are then the processing that we model. And the alternative is to say, I'm spanning spaces that are themselves temporal, so spatial temporal filter spaces, right? All frequency and space. And systems guys like that, like radio engineers, right? Radios are dealt with like that. You have a space frequency time, like what's the name for that little force, like Gabor filters would be an example, right? And then they try to make sense of that, and actually then there will be some static processing, like decomposition of a signal into components, spatial temporal components, right? I don't think that that's a successful approach. Doesn't really capture anything interesting, but it's always difficult to say something negative. Who knows, maybe somebody will discover something, but it's not been very useful in the past. Yeah. So in that sense, we're coming out on the other side, really saying we take these feature spaces static, and then everything time varying is the neurodynamics that does that. There was one more point I actually glossed over. I thought I had, yeah, this is the slide I glossed over. And so this is it's very clear in the motor case that, but it's actually true in both. In this picture, if I build these distributions from neurons, I said, I do that by using the tuning curve as the basis function. And so every neuron is actually smeared out over the population, right? So this is actually not an idea that the distribution we later have in the fields, that a neuron is in one location. Like on the cortical surface, you know, you would say, well, the neuron is in a location. Actually, in the cortical surface is also not quite true. I'll say something about that in a moment. So in this picture, the neurons are smeared out over the distribution because they distribute the whole tuning curve, right? So it's a bit more abstract than perhaps one first thinks. It's kind of interesting that our colleagues in Canada, Paul Zizek, they kind of smeared over that. They called it still cells. You know, it wasn't big, even though their method was exactly ours. Because it's a little bit shocking, perhaps, to realize that that's the case, but that's the case. And I think actually that is a very appropriate picture. So when you record from cortex, so I should say Dirk Janko has his lab right below this is his lab, one floor down, and he does now some kind of imaging technique. It's a way to how you can directly image voltage patterns on the brain. Voltage sensitive dye imaging and more recent stuff, and these images are driven by by membrane potential. So you can actually image the voltage in a network in real time. It's not a Bolt signal. It's not a hemodynamic response. It's actually real time voltage fluctuations through some mysterious process. And when you do that, then you realize, well, most of the membranes are in dendritic trees. So if you record from a location, it's not where the spike is generated, soma, or somewhere else. It's actually the dendritic tree on which the inputs come. And then you find that that there is very, very broad activation, because when you have a stimulus, you know, it actually is a very broad arborization that will be engaged in even similar, computing or responding to any localized stimulus. So that say it's actually somewhat special that we think that everything should be spatially defined by where the soma is, right? So this whole idea that neurons are localized, well, the soma is somewhere, but that dendritic tree on primary visual cortex, if I interpret the dendritic tree in terms of where spatially they are, so what the receptive fields are at the location where the dendritic takes input from, right? It takes input from neurons that are somewhere else and that have their own dendritic tree such that they have a receptive field somewhere else, right? Then the dendritic tree covers pretty much half of retinal space. This is true for the foveal area, half of the foveal area. So that's already population code. They are very broadly sampling from the visual area. So this idea that neurons sample something is actually certainly not in the sense of a point sample, right? It's much more appropriate to think of them as functions that sample a whole range. So later we will sometimes still say, oh, this neuron and that neuron, a location in the field, and I try to avoid that jargon, but it's really much more abstract than that. We should say an activation variable. Good. Yeah, so we're way over time. Yes? Yes, please. So we're almost, took much longer here. So see the first perturbation in the program, but we're already anticipating a lot of things that I'm going to say next. So about the kind of patterns of connectivity in the brain, I just want to make one point about that. So you all know that the neurons can be ordered in a certain way. I already hinted that from the retina to cortex, there's a forward network, a systematic pattern of connectivity in forward network. This famous work by Wiesel's student, I forgot the other guy, Gilbert and Wiesel, who figured out that the receptive fields are really made by this forward connection. There would be alternatives. You could think it's made by interaction, but they really were able to show that by intracellular recording from LGN and cortex oryzae, that the shapes of the receptive fields are caused by that forward connectivity. the one thing that I mentioned is Dale's law. Not everyone uses that term. Many neuroscientists think it's kind of trivial. So Dale's law is the idea that every neuron has only one type of synapse. So they make the same synapses with everywhere where they project. And it's also where they project. And that means, in particular, that a neuron either makes excitatory synapses or inhibitory. So that's why we can even say it's an excitatory neuron and inhibitory neuron. Right? So you never have a neuron that can do excitation for some synapses and inhibition for some other ones. It's also true with respect to which neurotransmitters, or even within excitatory, or within inhibitory, they're always of just one kind. And that will be important. The Amari equation, which we use a lot, actually violates that principle, because we're assuming that there is excitatory and inhibitory interaction. Now, I already said, it's much more abstract. The neurons are smeared out and so on, and we'll actually correct that in some cases. But that's the one thing that I want to point out. And then, of course, recurrent connectivity, just to make it, recurrent connectivity really means that there is some kind of a loop, right? That when you have, you cannot actually account for the activity by just knowing the inputs, because some of the outputs are also inputs. That's where you actually need time to disassemble that, to say that the outputs that serve as inputs are the outputs from some previous time. And that is actually concrete in the brain, how that works. Good. The last point about synaptic dynamics, meaning learning and Hebbian learning, and we all know fundamentally that's the mechanism. I have a little module on learning where I show our approximation of that. So I think you all know that there is this plasticity that is actually spike time dependent. So under certain timing, there will be strengthening of the synapse. Under other timing, there will be weakening. But we're not using that really in our work. We're just using a very simplified version of the Hebb learning rule. The Hebb learning rule, as you know, was postulated way before it was known how it's actually done. And again, as it was done, it was discovered that it was more complex. So the spike time dependency was not actually postulated initially. And we are not using that because we're not using spiking, right? So we're actually glossing over macroscopic detail. There's a lot of knowledge about the molecular mechanisms, also a lot of challenges about that, right? And the proteins needed for changing the neurotransmitters, the neuroreceptors to bring about plasticity are made in the soma. And it's gene expression, right? And then the proteins have to be transported there. And so there's much more to that, right? It's the synapse fires, but actually there's the whole cell needs to do something to update that. So it's quite complicated, and we're glossing over all of that, and of course, a whole field of research. Okay, so that's my introduction to neurophysics.